It's my great pleasure to introduce Roberto Colom. I have a story to tell you about Roberto. He, is a professor at the University of Madrid. He invited me over to give a talk in Spain a number of years ago. And to my great regret, one of the things that I love about Roberto and remember about my visit is the incredible collegiality that was offered me when I was there. Very friendly group of individuals and very welcoming individual himself. He is very collaborative, will work with anyone. The one thing to my regret is that in Spain you go to eat very late at night, and when they invited me to go to dinner at 10 o'clock, I said, well, I'll be in bed by 9, so I have to eat by 8. So I could not stay and adopt the Spanish way of friendship, so I aspire to that in the future. But I would like to introduce the great neuroscientist and psychologist, Roberto Colom, who will tell us about the current status of neuroscience research. Roberto.
Well, thank you very much for inviting me to this keynote address. I will introduce my talk using three main ideas. The first one is this. 25 years ago, this human being wrote a troublesome editorial note for intelligence. And in this note, there are three main ideas that will be a good point for my presentation. These three main ideas are this one. The first one is that sooner or later, all psychology research leads into the human brain. I strongly agree with this statement. The second one is that it's a question. Can psychometrics lead the way into the brain? And the third one is, as the decay of the brain begins, the study of intelligence is in danger of being left behind. I think we did some of this work that Rich put on this editorial note 25 years ago, but there are still many things to do. The second thing for this introduction is that this main idea that Professor Hunt put in this wonderful book about intelligence. At the end of the day, everything is in the brain. And the second one is that every expression of intelligence is due to actions of the brain. This is a main idea of what I am going to tell you today. And these two main ideas are behind of the main thesis of this presentation. This is the brain connection. What I mean with the brain connection is that what you can see on the left is important, the problems we are facing in everyday life or in the standardized instruments we use for measuring intelligence. And what you see on the right is also very important, but both things are combined here in the brain. All the relevant things that can lead us forward for understanding this psychological construct took place here in the brain. And this is what I mean by the brain connection. We need to go there and try to find out what is going on when the information outside and what we have inside is combined there in the brain. So let me, in the second point of my presentation, let me say something about how we dig, briefly, how we dig into the brain, the kind of methods we are using for trying to understand what is going on when people try to solve problems that require our intelligence. These are some examples. I am focusing my presentation on structural features of the brain. I am not talking about function. I will talk about brain structure. And we do many exciting things with the images we get from the scanner when we put there different individuals. We can use voxel-based morphometry, we can use surface-based morphometry, and all this kind of information is analyzed for trying to find how individual differences in this kind of brain features are combined with differences in intelligent performance. And by the way, note that I make in the low level of the slide interesting distinction between cortical surface area and cortical thickness. This distinction will be very important later on. This is one example of what we get when we correlate differences in volumes, regional brain volumes, and differences in general intelligence. We see on the left what happens when we compute the correlations between these two kind of signals, the brain signal and the psychological signal, and gray matter. And on the right, you can see what we get when we correlate differences in white matter, the connections between different areas, and differences in general intelligence. This is just an example. I will go quick for presenting many different data. And at the end, we will try to summarize the main idea of what I'm trying to communicate today. This is a typical finding. This is another example that goes deeper into the distinction between different brain features, and we see what is going on when we correlate differences between a cortical surface area and cortical gray matter volume on the left. In the middle, the correlation between differences in cortical thickness and cortical gray matter volume, and on the right, the correlation between cortical thickness and cortical surface area. The interesting thing here is that the correlation goes down. Indeed, there are some studies that found that the correlation between cortical surface area and cortical thickness is almost zero. And this is very important to speak about the biological nature of the G factor. We will see something about that later. And this is one example using more than 200 young people of what happens when we correlate differences in cortical gray matter volume, cortical surface area, and cortical thickness with IQ differences. We see that the spots showing correlations with these three different brain signals changes. This means that the differences in the psychological factor we are measuring here, IQ, correlates with different brain regions if we use different brain signals. It's not the same thing to correlate IQ differences with surface area and cortical thickness. And this is important for saying things about the biological nature of the general factor of intelligence. This is another example of what we are doing now for trying to understand from a biological perspective individual differences in intelligence. Here we focus our attention in the connections between different regions of the brain. And in the lower side of the slide, we can see the kind of fancy images we get when we try to get, nobody understands what is going on there. But we can do something for trying to clarify what we can get when we correlate differences in connectivity between different brain regions and differences in intelligence. Well, I am moving now after this brief presentation of some kind of methods for trying to get some idea of what is going on at the biological level. When we ask about differences in intelligence, I am moving now to the third point of my presentation, which will be a Gaussian tail. This is just to lower our expectations when we try to find something interesting in the brain about the intelligence construct. This Gaussian tail, I will go into some detail here, just to demonstrate the three main points you can see there. The first one is that the estimation, I will focus here in just cortical thickness, but we get the same thing for cortical surface area, the same thing. First, estimation of cortical thickness is not consistent across methods. If you use different protocols for getting information about differences, variability in cortical thickness, you get different things. And this is troubling. The second one is that there are important variations in the spatial pattern of cortical thickness cognition relationships among different surface-based morphometry methods. So the second one is two very troubling. And the third one, perhaps the worst one, is that within each, if we are using just one surface-based morphometry method, results did not replicate in matched samples. When you match carefully different samples of individuals. And this is just a scary slide. The many things we did for reaching these three main conclusions, many different things, very complicated things, and we can try to summarize what is going on in this terrible slide in this way. If we use three different imaging protocols for getting information about the differences in cortical thickness across the brain, we see that the maps change, and the maps change again in a troubling way, because from the right to the left, we see the CBET, brain suite, and CBM at the different protocols, and we see that the more the user intervention for trying to find the main landmarks in the brain, the better the approximation of the information you are obtaining to the information we know in post-mortem information. So the more the automatic the method for getting the information about cortical thickness, the worse we are approaching the real data of the brain, okay? And this is what happens. I will show you just two examples. We have more examples about this kind of, but the same thing that I will show you now in these two next slides. The first one is about what happens when we compare the correlations between performance on the Raven test in two groups of people perfectly matched in many interesting psychological factors and biological factors. And when we correlate these two kind of information, the performance on the Raven and variations in this instance in cortical thickness, we get very different results. The correlations are very different, and you can see here some examples. Here, you see a very high correlation, and there a very high positive correlation for this group B, and here the correlation is negative for group A. And this is just an example. Well, when we saw that, we thought, well, this is because the Raven is a very complex test. What happens if we use a very simple test that is related with intelligence? And we did the same thing for a a simple spatial speed test. This is much simpler. The phenotype here, and we will talk about more about the phenotype shortly. The phenotype here is much more simple, but the same thing, we get the same thing when we compare group A and group A and group B, and the correlation you see there, that there is a big difference between both kind of information we get for group A and group B. So there is no replication here. Okay, and why is that? Why we get different results for these groups match in a host of variables? I think there is an interesting answer, and this answer it can be found in this paper we published 4 years ago. And in this paper, we suggest that there are so many differences in the specific brain areas different individuals use to do different tasks. The Draven and the spatial test are two examples, that no one specific area stands out for cognitive performance on any given task at the group level. And this is a key message from my talk today. So it's difficult to find this consistency because there are huge differences in the way individuals solve a given task. Okay? Well, and now I am moving to the fourth part of my presentation, and this will be a big one, because in this fourth part I will try to find some answers to the question you can see there: where in the brain is intelligence? We have been working trying to find some answers to this main question for now for a decade, and I will try to summarize some of the main findings about this point. We try to find some information about key intelligence factors in this report that we published some years ago, and this research was aimed to find the brain structural correlates of the general factor of intelligence and two other main intelligence factors that were controlling for the influence of the G factor. So trying to isolate the brain correlates at the structural level of different intelligence factors at the latent level. And this is what we found here. We found, you can see what happens for the general factor of intelligence at the top of this slide, and what happens for crystallized or verbal intelligence in the middle, and what happens for spatial intelligence in the bottom of this slide. Perhaps the more interesting thing in this study was that we found a strong brain correlates for crystallized and spatial intelligence, controlling for the effect of the general factor of intelligence. And this means that not everything can be reduced to the G factor. This was one of the main messages of this report. But this study I just described briefly was done with a group of 100 people, young people, but they were clearly not representative of the population. So what we tried to do another thing with a bigger sample of 207 young people, children and adolescents. And in this case, the sample was representative of the population. So we asked, what happens if we do the same thing I just explained in the previous report with a sample representative of the population? What happens? And this is what we did here. In this case, we analyzed cortical thickness, no cortical gray matter volume, but the same thing applies to both kind of studies. This is on the left, you see what happens when we correlate differences in cortical thickness with differences at the latent level for school skills, verbal reasoning, and spatial reasoning. We get a lot of results. So the next question was, what happens when we remove the effect of the G factor on these three latent factors? Okay, what happens? We get the same thing that we get in the previous report. We still have some results for cortical thickness when we correlate school skills, removing the effect of G. What we get here was nothing. So we get nothing when we control the effect of the G factor for these three latent abilities. So for this representative sample, there is nothing left when we control the effect of the G factor. Okay? Well, I am moving on, and now I will present a study trying to find some commonalities or differences between the classical intelligence factors and some very important cognitive factors such as working memory, attention control, processing speed, and so forth. So the question here is a bit different at the two previous questions. And the main point here was, is there some commonality in the brain that accounts for the relationships between, say, fluid intelligence and working memory? Between crystallized intelligence and attention control, and so forth. Okay? Well, and this was the main finding. We failed to find correlations for cortical thickness, but we get some interesting findings for cortical surface area. You see there that there is an interesting overlap for fluid intelligence, crystallized intelligence, and working memory capacity. We failed to find any overlap for a fluid, crystallized, or spatial intelligence and the cognitive factors we analyzed in this study. And we mainly found that the overlap is on the right frontal lobe in broadband area 46 on the right hemisphere, and as you see there, for these three cognitive factors. So again, this result cannot be generalized to the population because this study was made using a sample of university students. But we found here that there is an interesting commonality in the frontal lobe. So we concluded here that the frontal lobe is important, not for fluid intelligence, not for crystallized intelligence, and not for working memory capacity. It's important for the commonality between these kind of factors, of cognitive factors. And now let me show you an example of why I said before that the phenotype we are measuring is important. We say that we are measuring intelligence with, say, one specific test, with a latent factor, with using IQ scores. We are using many different ways for measuring intelligence. We say we are measuring intelligence, but we are measuring what intelligence? And this is the main goal of this report. What happens when we correlate differences in specific abilities or in group factors or in the general factor of intelligence with cortical gray matter volume, cortical thickness, and cortical surface area? What happens? And this is what we found. This is a summary of this complex study, but it's evident from this image that we get a lot of results when we correlate a specific intelligence test that we get a smaller amount of findings when we move to the first factors, and that we go, when we go to the higher order factor, we obtain just 3 small clusters for the correlation, in this case, with cortical gray matter volume. So when we move up, we get less results with respect to cortical gray matter volume. So why is that? Why we are getting less correlations between both kind of signals? We guess that the main explanation for accounting for these results is that at the subtest level, we are having a lot of noise. We are having a lot of specificity, and we are getting findings that are not really interesting. The interesting findings, this is what we think, are obtained for the higher order information, because the specificity is washed out. I think I can handle this. Okay. So, may I have some water? Thank you. Well, now these are some examples of what we are obtaining when we analyze normal people. And now I will talk. I will present some findings using people with brain lesions. Okay, we use a method for analyzing a big group of patients, and we will present findings for 2 different groups of people to see if we can get some commonalities between these two studies, and if there are some differences. Thank you. So the method is quite simple. We just look how a specific lesion in a voxel, in a specific region of the brain, has some impact on performance on the intelligence test. And that's quite simple. You just need to identify the brain lesion and try to find if this lesion has some impact on intelligence performance. The first study was done with 241 patients, and as you can see there, these lesions were single, focal, stable, and we exclude patients with progressive disease or psychiatric illness. This method allows extracting causal. We think that we can say that if there is something broken there, and there is some impact on performance, we can conclude that there is a causal effect on this psychological factor. And the main question to answer using voxel-based lesion symptom mapping is which voxels preclude proper intellectual performance. And trying to find an answer to this question, we get this main finding. We get the finding that the general factor of intelligence is supported by the efficient communication between inferior frontal and superior parietal regions. So the main image in the upper side of the slide represents this main finding for the general factor of intelligence that we obtain controlling for all the specificities in the battery we use for measuring intelligence. And let me show you now an example of what happens when we move from the general factor of intelligence to a specific subtest. In the upper side of the slide, in the top panel of this slide, we see two specific subtests that shows a high overlap with the findings for the general factor of intelligence, the similarities test and the arithmetic test. These two tests shows a big overlap with the general factor of intelligence. And in the bottom panel of this slide, we see that picture completion and block design show a low overlap with the general factor of intelligence, which means that verbal intelligence and the working memory factor measured by the Weiss subtest represents quite well what we get for the general factor of intelligence, while for picture completion and block design, these two subtests, this perceptual subtest of the Weiss represents badly what we are getting for the general factor of intelligence in this sample of patients. And the next question we asked in this study was, is there a neural region whose damage uniquely impacts G? So there is some brain region that is especially important for G only. And the answer was yes. There is a single region in the left frontal pole in the Brodmann area chain. You can see there the white circle that is specific, and this is interesting, specific for the G factor. And when we ask why this specific region is important for the G factor only, and we suggested that perhaps this region is important for controlling the flow of processing. So there is something there important for the G, for what G represents at the biological level. That's the main result when we try to answer this main question. And as I said before, we did the same thing, the same kind of analysis using two different samples, trying to know if we can replicate the previous findings. So I will show you something about that, comparing the two kind of results. And this was done with Vietnam vets. And this is what we get with this, for this sample of 182 patients. And the main finding for the general factor of intelligence was pretty close to what we get with the previous sample. So all the interesting things are mainly for the general factor of intelligence, mainly focused on the left hemisphere. So the main things were happening there. And there is a high overlap with the important regions we found in the first study. And here is a comparison of what happens in this second study for verbal comprehension that saw a high overlap with G. And as I said before, the same thing happened in the previous study for similarities and the general factor of intelligence. And again, verbal intelligence seems important for understanding what we are obtaining for the general factor of intelligence. And this will explain some interesting things later. There is also a high overlap in the findings in the two studies for working memory. And as I said before, there is also a high overlap for arithmetic and the general factor of intelligence. So again, working memory shows a high overlap with the general factor of intelligence, again, on the left side of the brain. And we saw a very low, in this second study, we again saw a low overlap between perceptual organization and the general factor of intelligence. It's very shocking to find that the main findings for this factor that shows a very low overlap with G are on the right side of the brain. So this is difficult to explain. Why we have seen many times that there is a very high correlation between G and GF. But now, when we go on at the biological level, we see that for these patients, what is going on for something like GF is on the right side, and for the general factor of intelligence is on the left side. And the left side is also important for verbal comprehension and working memory. So there is something here. This is a kind of summary of what we get in the two lesion studies. For the first one in the bottom panel and the second one in the top panel. There are similarities for G, but there is a big discrepancy between what we get for G and for GF. So we ask next about the overlap between intelligence and working memory. We focus our attention not on intelligence, but on fluid intelligence. And here we get some pretty exciting findings. We ask for the correlation between different aspects of working memory and fluid intelligence. And we get what you see in the bottom of this slide. For general intelligence, we know that most of the findings are on the left side, while for fluid intelligence, most of the findings are on the right side. And this is what we get. I will show you now in the next two slides the two most important findings. But we again found here mainly a high overlap between what we get for fluid intelligence and spatial working memory, but not for verbal working memory. And there is a horrible overlap between fluid intelligence and the factor working memory that will be important to understand some things I will tell you later. There is a very low overlap between working memory monitoring and fluid intelligence. This is what I show you here, but bigger. There is a high overlap between fluid intelligence and spatial working memory, a very high overlap. And there is a very low overlap between monitoring working memory that in this case was measured by the M-back task, the different variations in complexity in the M-back task, 1 back, 2 back, 3 back. And you see that the overlap is very low. There is no any overlap between these two factors. Okay. I think I am going fine with the time. Okay, perfect. So now I am moving. You see that I have many things to say today, so be patient. Okay. Now I will tell you something about development, because we are trying to find if this very famous study is correct or not. Perhaps you remember that because it was very famous, because they found that there is a negative correlation between intelligence and cortical thickness in young childhood, and these correlations changed to be positive later. This was shocking, and we have been very skeptical about these findings. So we try to replicate that using a big sample of young people, children and adolescents. And this is perhaps the most important finding here. There is a negative correlation that goes there, that tends to be positive. And we try to replicate this, but I'm sorry to say that, but we being more careful than these guys. We choose carefully the individuals that we put in the different age groups, and we found, this is the main design. You see, the age range was from 6 to 21, and we split the group in young childhood, late childhood, early adolescence, and early adulthood. And this where I just will show the results for the cortical thickness. We get the same thing for cortical surface area. No negative correlation turned into positive correlation. All the correlations were mainly positive. This is the main finding of this new analysis using the same design that was used by So et al. So there is no any important change between early childhood and late adolescence. There is mainly positive correlations. And this is perhaps the most important finding here. When we go year by year from 7 to 16, and we compare what is happening with the superior intelligence group with the average intelligence group of people, of children and adolescents, we always get the same result. There is always an advantage of the superior intelligence group. There is no any sharp change across age, as So et al. Suggested in their analysis. There is always an advantage of the superior group over the average intelligence group. This is pretty consistent, and as I said before, we get this for thickness and for cortical surface area, the results were even stronger. There's always the same result. So it's according to these findings, it's not true that most intelligent individuals are more flexible. We fail to find that. And in a second study, also using this developmental framework, we try to avoid the use, the frequent use of IQ scaled scores, and we're trying to model changes in intelligent performance instead of trying to refer each age group to his own group. We model changes in the general factor of intelligence using this is the same time gap, and instead of controlling for G, we modeled G. So this is a big difference with the analysis I just presented before. And now we see there how as people age, they improve. They are more intelligent, and we can model that using this kind of latent approach. And the question here: what happens when we analyze the interaction between these G changes and changes in cortical thickness and cortical surface area? And we get these nice results. We see that there is a very big interaction between cortical thickness and changes in cortical thickness and changes in intelligence in the G factor modeled using this kind of framework. Okay, so we see an important interaction here. And this is a kind of summary of what we get when we analyze what happens from 8 years 20 years. And when we split the whole group in people with different G-scores, 75, 90, 105, and 120. Using this kind of approach, we get exactly the opposite finding that was reported by So et al. Here you see that the change is much more important for the low G group. It's much more important for the low G group, and it's change much. The change is much smaller for the high intelligence group. So just the opposite of what So et al. Reported, and that was very famous, and we failed to replicate that. Okay. Well, now I am moving to the two final sections of my presentation, and now we will say something about connections. Remember the point number 2 of my presentation, and now I will show some findings we are getting. These are brand new results. They are not yet published. We are working on it, and the first one is based on the PFIT model. We ask Rich and Rex to nominate the regions in the brain that was key for the PFIT model. And from this information, we analyze the connectivity, the structural connectivity between these main brain regions. We divide these regions in the 4 stages that the PFIT model highlights. Okay, so we analyze the connectivity between these brain regions, and the way for doing that is it's a bit complex. It's an engineer thing, so I'm not interested in that. Just we use several computations for achieving this main conclusion. And this is a main finding of this analysis. You see there that there are some bars for stage 1, stage 2, stage 3, and stage 4. And it's clear in this slide that the connectivity in this network defined by these brain regions, the connectivity is higher for the nodes, the regions in the brain for the stage 2 and stage 3. And stage 2 and stage 3 are, no surprises, the frontal and parietal brain regions. Okay, so there is some interesting support here for the PFIT model. The connectivity in these regions is high. And from here, we correlate the global efficiency and the local efficiency of the connectivity in this PFIT network with differences in 3 kinds of psychological factors: fluid intelligence, working memory capacity, and speed, to see how differences in connectivity in this network are related with these 3 psychological factors at the latent level. And we found an interesting thing, that the biggest correlation was found for working memory capacity, the value around 0.50, that in this kind of studies is very high. And the correlation for fluid intelligence was in the expected direction, but not significant. And for the same thing for processing speed. The main conclusion from this study is that differences in working memory capacity are related with differences in the global and local efficiency within this brain network. Okay. And now a very fancy study that still not published. It is a brand new study, a very complicated study that I will try to summarize to you in 3 minutes. So I will try to do it. Okay. And the name is terrible. It's a multivariate distance matrix regression analysis. It's not great. So for short, MDMR. And this is the way, the main step for computing this kind of complicated analysis. I try to explain in the simple way. We are looking here, we're trying to answer the next question here. Is people more alike in their connectivity in the entire brain, more similar in their fluid intelligence scores? Okay? This is the main question here. And we try to analyze that bottom up. No hypothesis, no nothing. Just see what happens. What happens when you analyze the biological information and the psychological information at the same time? What we get there? This is the way for doing that. We work with weighted structural connectivity matrices that we try to visualize what is happening here on the right. Impossible to see anything. These are the regions we use. There are 82 regions here. And we began with more than 3,000 connections. We analyze all this information and try to simplify this information in an inductive way. And this is what happens when we study the information using simulations and real data. Okay, in the top panel, see what happens when there is no any relationship between the similarity in the biology and the psychology of the individuals analyzed on the left. And on the right, you see that there is it's a kind of graduation from the main diagonal to the angles. There is a kind of orange, and then it turns blue. Can you see that? Something like that. Okay. And this is what in the bottom panel, what happens with real data. What happens in our data set. And this is the main finding. We began, remember, we began with more than 3,000 connections, and we end with just 36 connections. Just 36, which means that people more alike in these 36 connections are more alike in their fluid intelligence, crystallized intelligence, spatial intelligence, but not in their working memory, processing speed, and attention. Okay, this is the main finding that we get here. Okay. By the way, you see there in the top panel the left frontal pole in the brain again. Okay, and now I am moving to the final section of my presentation with two main things. The first thing is an answer to this question: how can we improve our brains? And I use this sentence again of my dear friend Rich, that recognize that the ultimate purpose of all intelligence research is to enhance intelligence. So I will show you just one example and move to the final stage of my presentation. Remember when we talked at the beginning of my talk about cortical thickness and cortical surface area? That I told you that it was an important distinction? Yes, no? Well, it's interesting to know that the genetic correlation between cortical thickness and cortical surface area is almost zero. So these two brain features are independent. And this is not a result of our group. It's a result of this group that was led by Bill Kremen, and they found an interesting parcellation of the brain for cortical thickness and cortical surface area. And among other things, they found that the correlation between these two kinds of parcellations at the genetic level was 0. And this is important for the unitary nature of G. We are working with this group trying to find if these regions of the brain, based on the genetic information, are sensitive to cognitive training. Okay? So this parcellation was made with free surfer. That is a program for analyzing images, and we needed to convert this to the MNI space. So we did here a very difficult work for transforming this information, but we get finally interesting results for both cortical for cortical thickness and for cortical surface area. I will try to summarize the main finding for these two kinds of information. Okay, this was the main design for this study, a typical pre-test, post-test study. And this is what we get when we compare the group that completed the training program with a control group. And we mainly found an interesting result for the temporal lobe for both indices, for cortical thickness and for cortical surface area. There, in the top panel of this image, you see that the overlap between both is mainly in the middle temporal lobe. Okay, we get also findings for thickness and for cortical surface. So what this image indicates is that there is a difference between the control and training groups in these main regions. So there is a statistical significant difference between both groups. But perhaps the most interesting thing we found was using this parcellation scheme is that people that was more sensitive to the training program was people of the training group showing lower intelligence scores in the pre-test. I think this makes sense. People with a low intelligence level was the ones which show beneficial effect of the training. Okay, and this is what we see here. Okay, we are working now briefly. We are working now in a huge project that tries to overcome many problems that are found in this training staff, and we are working with a very interesting group trying to analyze 2,000 individuals, just to escape the critics about small sample sizes and so forth. And in this project, we are combining cognitive training, physical fitness, nutrition, and brain stimulation, trying to find which is the best combination for, take note of that, improving fluid intelligence. This is the main goal of this project, trying to improve fluid intelligence. Well, this is the end. I promise that. So I will say a couple of things about the future. I had the opportunity to explain what I mean by the future in this kind of research, in these two short reports, and mainly what I can say about the future is that we need this kind of stuff. We need to get refined information of the brain to improve our way to analyze the images we get from the scanner. And this is an interesting example, big brain, that is currently used for refining this kind of protocols. We can also move to another kind of technologies, and I think this is a promising one, trying to analyze in real time how brain cells are changing information. We can do that now, and we are improving doing that. We can also rely on these kind of frameworks. I love this one, by the way, and trying to analyze how complex systems work. This is, as the name suggests, complex. But I think that Daniel Bassett is doing a very good work trying to understand what is going on the brain, the connections in the brain in a dynamic way, and we are improving also in this line. And perhaps this is my favorite. It's a Japanese group that is trying to understand how the brain works interacting with the brain. And this is a very fancy thing that they call decoded neurofeedback. And I recommend you to go there to see the fancy images of a human being with an exoskeleton trying to do something and send information using the brain waves. That's really interesting. And I think this will be an interesting approach for improving our knowledge about how we use the brain for solving problems. And also, we also need to combine the information we are accumulating across the years. This is, you know, this one. I think you know this one. And this is an interesting meta-analysis of the information we get about the brain structural and functional correlates of intelligence. The interesting, perhaps the most interesting thing about this meta-analysis is that there is no any overlap between findings for the structural information and the functional information. There was an empty brain when they combine both types. We don't know why, but there is no overlap between the structural and the functional findings. This is also a very interesting source of information, neuroscience. And this is also another web site for obtaining information about brain function. There is a lot of information there, but we need to get some idea of what is important here for understanding intelligence differences. And we also need to combine genetic information with brain information. And this is one example that probably you know, Enigma. So, finally, I am arriving now at the end of my talk, of my presentation. And remember that we began using this sentence from Professor Hunt. And my main message after all this information is that a deep understanding of what is going on in our brains when the genotype and the environment are integrated, and this is what I mean by the brain connection, will set up the framework for a deep understanding of everything else. Thank you very much.
I once did an fMRI structural study with IQ, and the neuroscientist I did that with presented some data. He was very thrilled about it, wanted to publish it, and then a couple of weeks later, or somewhat later, I don't know, he wanted to redo the analysis and was unable to replicate his earlier findings. And he said, no problem, we'll do it again. And he did it again and found something else. Are you worried about this as well, as much as I am?
I am.
Okay.
Absolutely. So I share your concern, yeah.
So, given the smallness of samples, do you agree with me that it's very good to always, given the fact that given the lack of knowledge we have about many relations with the brain and IQ, because it's an exploration, basically, given the many statistical issues involved, would it not be always good to have an independent replication sample to verify results to really make sure?
Yeah, that's why I was interested in explaining to you the MDMR method that I think is very promising for trying to cope with this issue you are pointing. Yeah.
As well as collaborating, I guess, because having other labs.
Enigma is a perfect example.
Yeah,
That's one of the main messages of my talk, that we need to work together as much as possible. We are failing doing that, by the way.
It's difficult. I mean, in the neuroscience, it took them a while in molecular genetics, but I think that what happened there was interesting, in the sense that you had different camps competing like crazy against one another, doing the first GWAS of whatever condition it was, and then Team A winning, scooping Team B, and then Team B having just collected data, reviewing work of Team A, and then at the same time saying, well, I'm sorry, we just did that same study, we couldn't replicate, we get completely different results, and then editors saying, hey, why don't you team up? And then they teamed up. So it's basically, you need a bigger scale, you need international collaboration. I hope that maybe within ISIR or other neuroscientific circles we could combine forces.
I think we have some role in huge projects such as the Human Brain Project or the brain here in the States. I think we have to go there to try to collaborate with people that's now doing something about that.
I'd like to follow up on that and perhaps get some comments from some of the employment psychologists here. You know, it reminded me of the great confusion not early, but in the 60s and 70s about the inconsistent results in predicting, in different abilities or intelligence predicting job performance. And what it came down to, for the most part, not completely, was statistical artifacts, unreliability of measurement, sampling error, the small samples, and also, very importantly, restriction of range. Because your correlations are going to drop if there's a relation actually there when you're using only university students, for example. And so I'm wondering if adopting some of the concerns and strategies, or just looking at it through those eyes, not all the studies, but would help you bring some order, some consistency. I know Paul Sackett, and we have two representatives up here, could maybe offer some opinion on that.
I agree with that, and that's why I began my talk with the suggestion by Rich about using psychometrics to collaborate with people doing neuroimage.
I'd also like to follow up on these points, especially Yelte's, with the reproducibility of these findings. I mean, one can frame that as a problem or a concern, but it might also very well be that that is a fundamental property of how G or intelligence in general is situated in the brain, because, as you were saying at that point, it might very well be that people use their different brains differently to reach similar intelligence results, so that we have a major case of multiple realization of intelligence in the brain there. My question to you would basically be, do you have any idea how we could get closer to that and try to model that in a way? I'm thinking about, for example, if we have samples of individuals that are very similar in IQ and look at their brains and try to find if we can find configurations of the brain that explain the same IQ level in different people. Isn't that some interesting way forward to get around this problem?
Yeah, that's a difficult question, but I think we need an answer for that question. And perhaps one way for trying to clarify the field is to avoid now using group data. try to find out what is going on with each individual, and try to get some information from there, but not go directly to get average or something like that. I think that's dangerous and very confusing. That will be my guess, my prediction for going forward.
Thank you, Roberto.